Thank you so much for having me. This has been a fantastic workshop. So I worked on math, and I guess I should say theoretical computer science a while ago. I would say, I run an experimental lab, so it's really a pleasure to get to talk to many of you, and I hope I'll tell you some interesting things that will stimulate some good discussion, or maybe help formulate or think about some problems at least. Yeah, and as best I can going forward. All right, so I want to talk about, when we're sick, we take, don't worry, you don't need to know any chemistry. These are antibiotics, and don't quiz me. But this is one of the technological, I would say, sort of most impactful technological achievements of the 21st century are really understanding that we could fight infection with molecules, and the idea that infectious disease is the most important killer of humans is now much in the past, although many people would tell you more work is needed in the 21st century to keep that going. But it's humbling to think about the fact that really these molecules are just sort of a perturbative effect in this very complex and ongoing war, because what's really going on, this is my singular favorite YouTube video. It's taken in 1950 on a film camera. That's a neutrophil, so it's a white blood cell. It is chasing down a bacteria. That's that tiny little thing. Those big inert objects are red blood cells, and you can see this is the trenches of infectional war, right? So this is nothing like a small molecule. It's a machine, and in fact, the immune system is this whole army, right? And it's totally different. So if we were to come to a pharma company and say we want to build something like this, I mean, we have immunotherapy, but actually build something new, they think you're crazy, right? This is still really difficult to even envision. And I guess this is really kind of a piece of gel, right? Or even a bag of liquid. But what's inside is this network, this distributed system of molecules. And this is a small part, right? This is a network that is telling you, essentially, this neutrophil is chasing it smells, right, a molecule. And that molecule triggers a cascade of reactions that are eventually going to drive sort of physical changes that cause the cell to move and to form and chase this object, right? So it's a piece of Jell-O with a network inside. And so the question that we ask in my group is, how can I build something like this, and how can we start to build new materials? And I'm not going to show you a working neutrophil, but we can sort of think about this as a general problem. And I'll phrase it this way. It's not really a math equation, but I have some gelatinous soft material. I'm going to add to it some molecules that do things. So I'm going to think about a gene network, and mathematically sort of signaling networks and molecular genetic regulatory networks actually look similar. So some molecules that are coupled and have interesting physical interactions, and I'll try to describe that. And this is a large language model's vision of what should happen when you make a little robot. I do not actually have that, but it's kind of strangely adorable. So this is a problem I want to tell you about today that we've been thinking about for a while. So we spent a lot of time building toys. Actually, maybe I should go back and say a couple of other things, which is that we spend a lot of time building these toys, so we build things to move. So this mechanism that Govind just asked about, we build things to sense. So right now I triggered pressure. Also thought about building things that can sense light or specific kinds of molecules. Then we spend a lot of time building these genes that can connect different kinds of actions, whether it be moving, growing, releasing a drug, to specific information, and make smart decisions about when and where to do this. And I should say, we're not probably, and we can do a lot of this by building molecules. So I'm practically minded in the end. My hope is that, I don't know if I can build a neutrophil, but we can really start to think about maybe building some devices that would be pretty useful. So how many of you guys heat your house with an open fire? Okay, no one, good. All right, so for a while people were heating with a coal fire, right, that you would sort of stoke. But now that's also crazy, right? I use a thermostat, right? It turns on my heater when I need it and not when I don't. If you take a drug, unless you have an insulin pump, almost certainly you do not use the open fire method, or maybe this mechanism more like a coal fire. It's sort of guiding release, but there's no feedback between whether you need the drug or whether you don't. You just have to keep taking it and hope that you're taking the right dose. And we spend a lot of time studying this, right? So a device that can actually measure whether there's drugs or whether you need them would be extremely useful. So that's one of the problems we've been thinking about using these kind of robots, not as toys, but as ways to repair things. So I learned recently that the problem of corrosion, so this is a corroded model, costs the world 1 to 2% of its total GDP. So it's a very expensive problem. So that's one problem we're really interested in. And then also trying to think about how we can build sensors and sensor networks with some of the ideas to actually look at more complicated physiological state, like immune monitoring. But what I want to talk about today is actually what it's going to take to solve some of these problems, and that's to think about how to put these parts together in the right way and how to use molecules in a system to make good decisions. And often these decisions require coordination. So molecules are inherently local, or they're sort of mixing around. And so we have lots of what I conceive as interesting programming problems. So this is a model problem. This is a problem of proprioception. So proprioception sounded to me like a big word, but it basically is just your sense of balance, right? So whether you're upright, whether you know sort of what the physical world looks like around you. And plants also have proprioception, right? They can know sort of where to grow with respect not just to light, but also to their physical environment. And in us, right, proprioception is actually our balance, but also this sense of integrating lots of different signals. So for example, where your muscles are tensed or contracted, where you have sort of pressure at different points in your body, and where you have tension, right? And you can use all that information to get a really good sense of sort of what the physical world looks like for you, right? So for example, like I'm standing on the ground. I'm going to feel really weird, right, if there's like force around me in some other way. And so one could argue, if I have a robot, soft or not, it probably needs a sense of proprioception. This is a not great robot, right? It's not mine. This thing is a school project, but right, it runs into a wall and it doesn't have a sense of how to move. Right? This is a basic problem that you would run into, and we did run into in thinking about how to program a robot to move around, because it needs to have some sense of where it is. Okay, so how could we do this? So what we're going to do is we're going to think about our Jello, and I'm going to think about an array of sensors, and it's just going to be an array of posts. They're little, and each of these is going to be about the width of a human hair, so about 100 microns in size. And we can embed this now in a bigger piece of Jello. We spent a while printing these kinds of structures. That's what this paper is telling you about. This is a picture of the gels. We've been studying these. They're nice to look at without the other pieces of gel around them. And here's our problem. Okay, so let's say I have a sort of array of sensors. What I'd like to know is how I can tell what is pressing on them, right? So it's something that's bigger than an individual sensor. How could I create consensus? So I could have, for example, some square pattern of stimulus. And here I can talk about mechanical. I could also think about this problem as a light simulation problem. In math, you can just say essentially this is the set of inputs, right? And then you want your output to encode the shape of your input, and you want everybody, all the elements, to be producing this output. And if I produce a different pattern, right, I want a different output, right? So this is the flavor of this problem, right? I have some Boolean. You can also formulate this as a continuous problem. And then you want input and then a steady state. Right at the end, you want the dynamics to converge to some single output produced by all of the species. Okay, so how might we do this just with molecules that I can build in the lab? So I guess I can phrase it this way, right? Okay, so first of all, this problem is hard from the perspective of an engineer. Even outside of the design, biomolecules are really bad at computing, right? So if you're used to computers, right, a gigahertz computer is doing an operation every 10 to the minus 9 seconds. The fastest, one of the faster operations in humans is like 30 milliseconds, right? Biochemistry is happening on the scale of minutes. So the kinds of, reactions that I'm talking about, big changes in state are happening on the scale of hours, right? So I would like to have a response as fast as possible. And this is my favorite problem. Cells are really bad at encoding information in the form of concentration. So this is a really beautiful piece of work where they measured the variation in proteins concentration of all the proteins in a yeast, so this model single-celled organism, and they measured these among different species, and they can look at the coefficient of variation of the noise in these expressions as a function of sort of total concentration. What I want to call your attention to is this number, 10 to the 0, which otherwise known as 1, right? So the variation here is, and this is 0.1, is incredibly high, right? So you have a lot of molecules, but they're each really lousy reporters for any kind of information. So this is really a kind of yucky problem, and the only thing I think you have in your favor is that you have a lot of them, right? So how do we use that to think about? And I don't know the answer, but I want to tell you about what we've been thinking about. All right, so lots of questions about biomolecular computing have in fact focused on this. So we're going to make a test tube of lots of kind of molecules. They're going to interact, and we're going to encode state as a concentration of different molecules. I have lots, so I can encode state as some vector, right, in some way. Although you quickly run into this problem that if you have now a chemical reaction, that every species can interact with every other one. And molecules are kind of all like each other a little bit, within, I'd say, 6 or 7 orders of magnitude. And about, as I said, there's a lot of variation in concentration. So this gets hard quickly. We can contrast that with the scaling Moore's law gives us of having lots of circuit pieces where every piece of every transistor goes in a place. I can make more and more computing operations by just making everything smaller, not to mention faster, and I can now make billions of transistors. I can make a really big circuit. If I use this method, right, I get lots of parallelism, but I run into lots of crosstalk really quickly. And in fact, there's some really beautiful work by Margaret Johnson and Gerhard Hummer that I did not put on the slide, sort of showing that about a few thousand molecules with proteins and the way we know how they work, you run into this problem that you will get too much crosstalk to do anything useful. So in fact, this is a challenging limit with the chemistry that we know how to build. So how do you solve this problem? Well, there's not a simple answer, but one point. So this is a rainbow. This is this beautiful experiment where you can dye neurons in different colors to trace lineages and also see tissues, is that you use both molecules in some local environment, but you also make some kind of structure, right? You build a tissue. And we can think about this. In fact, there are new tools. They're really beautiful. This is a technique called CODA that can actually take old pathology slides and reconstruct the anatomy of centimeter scale tissues down to the single cell level. So we have lots of information about how anatomy constructs these sort of atlases at that scale. And our intuition is that somehow as engineers, we need to combine sort of chemical reaction idea of parallelism with some kind of structure. So that's how we ended up, in part, with this array. And so how might this work? Well, cells talk to each other using signals, right? So this is a diagram from Khan Academy where cells will release a molecule. So they'll send out, it's read by a receptor, and the cell says, I'm going to update my state. So this is a nice way for cells to talk to one another. And this is generally the case, and then inside we can think of cells inside as having some kind of logic response. And we can then envision this idea that we have these little devices that are sort of receiving signals from the outside, sending signals, and doing a little bit of logic. And that's a model that we wanted to use. So here's our sort of vision of how this might work with our array of posts is we have what's called a broadcast computer. So my cells are going to send signals. And the idea, and they can send any of a library of signals, is that these signals have some radius, right? I'm not going to, like a radio station, I'm not going to broadcast to everybody all the time, because what's the point of geometry if I do that? But I'm going to broadcast to some radius, and I'll show you how we've been working on doing that. And the idea then is that I can tell a range of my neighbors what to be doing, but I can tell them a lot of pieces of information, and all these channels and their radii can kind of overlap, right? So now I have a computing model. And if you think about this for a little bit, you can see that this computing model actually looks, so we have things sending signals. I have this sort of simple computation with nonlinearities inside. And in fact, you can see that you can map this to a neural network, right? It could be feed forward. It could also be recurrent, depending on the architecture, if you could program what each cell sends how it integrates that information, and how far that signal goes, and how it, and maybe in terms of integration, what concentrations of inputs at what level are going to tell you what to do next, right? And so you can change what you send based on this. And from this, our question is, could you build consensus? But also, could I build this? Is a sensible model that's chemically relevant to an engineer? So, all right. So I want to tell you a little bit about how this might work. First of all, we do not extract genes from organisms. We build them. Essentially, this is what's called the fundamental dogma of molecular biology. So the idea is DNA encodes some sequence that's transcribed into RNA that makes proteins that then tell DNA what to send. As an engineer, I don't want to be bothered with all of that trouble, so we thought we might take a shortcut and just do this. So we have a permanent code for each of our cells, which are these posts, and I'll try to get to some of the things that our cells are lacking. First of all, they don't reproduce, they don't grow, they don't manage metabolism or any of those things. They're only designed to talk to each other, right? So, and do little computations. But for our purposes, the computations are essentially just making signals and reading them and updating their state. And this makes engineering problem much easier. So I could buy a bunch of enzymes that people make in the lab. They've gotten cheaper because people use them to make mRNA vaccines, so yay. And we make DNA that we can buy from a factory for very cheap.
How big are your genomes?
About 80 base pairs.
80 base pairs.
Mm-hmm. Yep. Okay, so maybe more specific, this is a diagram. So this is our 80 base pair thing. It's got this rectangle that's called a promoter. That's where this RNA polymerase binds. T7 is another, it's actually a bacteriophage that infects E. coli. And we have another molecule called an RNase, and we have RNases that basically chew up signals. And that's super critical, right? You need to make information, but you also need to erase it, right? So those are our two enzymes, and then we have food. So our food are nucleotide triphosphates. These are the ingredients that are put together by polymerase to make RNA. And so this, you can think of this as our gene. It's kind of like a switch. And the idea is the following: has two states. It's either making RNA or it's not making RNA. So you can think of it like a transistor. We're going to gate it. It's in its on state or its off state. And we can make lots of these. They can, unlike a transistor, they're either making a particular type of RNA, which we can choose by changing what we write into this gray region, or, but the mechanism is sort of the same. And the difference is that what's happening inside this rectangle is a little different. So there's a hole up here, which this enzyme no longer recognizes that motif, and it's happy with this one. Okay. Right. So I'll just try to tell you a little bit about how this works without getting too deep into the biochemistry. The idea is, if we want to change state, we can write this message. This message can come bind over here to this toehold, which means it will pull this strand off, and suddenly this now has gone from full, happy, to broken. So this RNA is sort of a repressor. It acts as, basically changes this genome so that it can no longer bind. And we can do this other trick that's actually really beautiful, and not in the work of Jong and Kim, whose references on the previous slide. The idea is that we can control whether this happens with stoichiometry, because the idea is what we're going to do is have a bunch of additional sort of molecules up here that this one will bind to first. So this is a threshold, this stoichiometry. And once I have more repressor than I have this activator, and I can make different concentrations of these inside, this repressor can come and that it will bind. But if I have less, all this RNA is going to get bound up here, and eventually it's going to get chewed up by this RNase before it can ever go over here. So in fact, I can get this threshold, and I can tune this threshold very nicely with the concentration of this activator. I can tune how much I make by tuning this. And I can get this very high nonlinearity because I'm going to use a lot more activator than I am of this. So essentially, I get this curve where I saturate. And if I have more RNA than the sum of these, I am totally off if I'm in this range. And only in this range is sort of my transition region. So I have this very nice sigmoidal curve that I can write by changing my recipe. Okay. All right. And in general, you can see that, in fact, these have a sigmoidal response to a specific input. they make graphs that are compositional. They take RNA as input. They produce RNA as output. We can tune them. As I said, we can tune their switching. And also we can change their dynamics so they can turn off and on because we have this sort of constant degradation of the current signal. Okay. Any questions for you?
Trying to understand how I would program. So like.
Yeah. Yeah, so I'll show you an example, but the basic idea is I'm going to make a set of templates with specific inputs and outputs. So those are my nodes, and I'm going to choose the concentrations of those and their activators, which are going to give me these transfer functions, and that's going to give me this graph. So, yeah. So, okay. And then maybe I should just also say that one can show that the dynamics of this are actually, so these kinds of networks, are actually the dynamics at some level of neural networks with sigmoidal activation functions, right? So you can think about, in some sense, there's a mapping between the set of networks we can build and the set of artificial neural networks with sigmoidal activation functions. And one can also note that this is true to an extent if you sort of squint for natural gene networks as well as signal transduction pathways. So that's one thing you remember from this talk. I think it's a beautiful fact. All right, so here's an example of a graph we built. So this is a set of nodes that each have their inputs and outputs, and so we're drawing the connectivity. And in fact, you can see, so this is so these blunt arrows mean this one turns off the other one. And the idea is this is a flip-flop or a bistable switch. So A represses B and so on. So there's two stable states of this. And we can measure each of these signals with these little sort of monitors. So they're just producing fluorescence rather than RNA for us to read. So we can see what's happening during an experiment. And you can see that there's state on and state off, right? And then if you add some a stimulator, so this switch molecule, you can switch state. And there's a program that these turn on when they enter one of their states. And this is a pulse generator. So in systems biology, this is known as the incoherent feed-forward loop. But the idea is that when you enter this state, you turn on these two genes. That turns on this gene and also this gene. So initially, you turn this one on, you turn this one on, this turns on. You turn this one on, and then this one turns on. But this one turns this one off. So what happens is you initially turn this on, but later you turn it off. And that's what you see here. So when you start in state 1, you achieve this pulse. If I switch states, now I achieve the pulse of this other network. And I can do this in reverse, for example. Okay. So these are the kinds of little programs we can build. And they're not actually that terribly little. We actually have about 15 different nodes that we can build. There's lots of things that don't work. But I can comment that we have 15 inputs and 15 outputs. That's about 225, and actually I think we have more now, different components. And depending on how you count, right, that's maybe the size of a medium-sized virus. Okay, that now we can write our own genome programs, not for viruses, but for our Jello. Okay, so what we can do is actually take these, and we just sort of hook these genes up to the polymer network, which is what's holding this Jello together. It's a network of physically connected polymers. We're just going to bind the end of the short piece of DNA to our Jello. So that's the sort of software in our posts that's doing something. It's also the software that produces an input signal. And we can print not just regular arrays of structures, but actually multicolor arrays. So different posts can have different structures. This is something where a student just took an image and had the computer program it. Essentially, we just print, and we have a device that squirts different liquids, so kind of the way you make chips. So we can make arrays that have interesting conditions, which I'll show you in a minute. Okay, so here's our idea. So how do we get these posts to talk to one another? Well, if each of them are making RNA, what we're going to do is send that out. And I'll start by telling you that. And then the question is, how do we actually get this RNA signal to do something useful, such as turn these off or not, depending on where they go? All right. So here's the basic idea. So we're going to make RNA at this post. It's going to diffuse away, so Brownian motion, but at the same time, it's going to be degraded, right? So what you get in the end is a stable gradient of RNA if you leave it on for a while, like this. And we can do this by just putting these DNA inside these posts. And so that leads to this dynamic. So you have RNA production inside the post. and you have degradation of your signal everywhere at some rate. And what that gives you is a set of PDEs that are not terrible to solve, although the solution is yucky, depending on the dimensionality that you look at. But what you get is this sort of curve that looks a bit like a Gaussian. It's actually a set of Bessel functions. So what we did to measure this, though, is we made a gradient generator, so this little post. We made a second kind of gel whose goal is to measure the RNA. So essentially, it's just lighting up depending on how much RNA there is. And then what we can do is track the formation of this. It's actually sort of a field, literally, you can think about over time. And we can compare that to what the PDs would tell you. So essentially, if we let these, we can couple the production of the gradient to the measurement of the field by these sensor or reporter posts. And we can see this looks a lot like what we see. There's a little bit of magnitude difference, but it's not terribly bad. Okay, and then what we can do is ask, well, is this work in parallel? Because otherwise I don't really care. So we could take a bunch of these posts. We could let each of them make their signal. We can make a bunch of generators to track which post is listening. And we can do an experiment in which we use these posts to make signal and everybody else to measure it. And that's, in fact, what we see. So this is data from a microscope. Looks fairly reasonable. And then we can do this with lots of different shapes. So we can program. So what this is telling you is, yes, we can make an array of sensor posts. And when they are on, they can tell nearby signals. But critically, what this is telling you is even if you wait for a while, they're not telling these guys that are far away nearly the same information as they're telling their neighbors, right? So there's actually this locality of communication, and it's what I predicted in my model, right? And and we can do this in general. Okay, so great. We can talk to each other, but can we listen? So the basic idea is we're going to say, I want you, if you send me a signal that's high enough, I want you to do math, but the first thing is I just want to know you sent it because I want you to turn off your signal making. And this problem biochemically was not too hard, but actually figuring out how to do a good job with building sensor networks that could actually do anything resembling distributed information processing took a little bit of thinking. And so here's the idea. So we actually have this sort of chevron structure, which says this is my input, and I'm going to make, this is the message I make. So this one makes message 2. It's going to send message 2, and anyone who has an input is going to turn off. But there's also this sort of half chevron here, and this is that threshold I told you about. You absolutely need this threshold to get reliable and sort of, for us, comprehensible distributed communication. So what we wanted is, as I said, cells will respond when near and not when far. And what we found is you absolutely need a threshold, and it turns out this is about 100 times more threshold than we have signal to get what we can see, this response where you turn off if you're nearby, that's this over here, and turn on. If you don't have a threshold, you're going to get this sort of this curve like this, so you have no region of influence. You sort of have this analog behavior. And if you're not careful about how you build this threshold, you get this bad behavior. And so our hypothesis was if you can get what we call the sort of distance response curve, that how you respond to a neighbor has a direct relation to this, that we could then program. So to ask this, we took all the rates that we measured and the pieces that, well, I should also say this actually works. So we built 2 cells, and we put them in a gradient that we made with a microfluidic device. So same exact chemistry, but they were just receiving different amounts of RNA in the same pot. And we can measure this, and we can see that only one of them turns off, which makes it light up. And so that's this notion that this cell and this cell are exactly the same. They're in the same bath, but they're in a different place and getting different local information. So that tells us we can really build these. Okay. So the last thing I can tell you is we have a big array of these. So we made about 15 different, actually it's 20 different messages. I should say 15 of them passed our performance bar. And this orange is telling you here that this turns on, but if it gets a different signal, we tested a bunch of random ones, they don't talk to each other. So essentially, if you get the wrong information, you ignore it. If you get the right information, you switch, turn off, right? Okay, so here's a network we can build. So we have essentially, we want this node, this message to be sent to this one, and so on. But we're going to do this in a linear array. And so now what I'm going to show you are simulations where we take all the data we have, and I want to show you we can propagate information down this line. And the idea is that G1 is going to send a message to turn G2 off, and so on. And now I have a G1 over here that should only be talking to, so this G5 should only talk to this G1, not this one, right? And then that one should talk to this G2. And what that should give you then is this notion that this one is on, off. You can see that that one means this one should be on and this one should be off, even though, again, they have the same chemical makeup, right? But just different neighbors. And that's what we see in our PD simulation. And in fact, you can see that they tend to do this in a cascade. So we set this node to be on, and once we do it times 0, and as we do, you can see node 2 turn off, then node 3 turn on, and so on, and so forth. Okay.
So what are you doing exactly in these simulations?
So we just set all of these to be on, and then we see what happens when we let them turn on. So we let them go. So that node 1 is constitutively on, but all the others, once we set their initial condition, they can change.
Well, I think what I really meant is kind of what's under the hood in terms of, are you solving the PD?
Oh, this is COMSOL.
Yeah, okay, fine.
Okay, so it's a reaction-diffusion simulator.
Yeah, that's what I thought. It's
re-encoded. I mean, I can talk about the boundary conditions, et cetera, if you want, but yes, that's it. There's nothing fancy.
I just wanted to know,
this is a standard set of equations.
Yeah, no, it was that you were solving the PDs on.
No. I don't know how to solve these. You can solve the single gradient, and we've done that in different dimensions, but this is easier to see what we're doing. In fact, because also, well, this has a steady state, but I'll show you there's others where we're interested in the dynamics, so then it becomes really disgusting, because also I made these square and all sorts of other things that you wouldn't really want to solve that. I mean, maybe you would, but I don't. So, okay, any other questions? Okay. All right, so here's another example where I can have a network where, for example, this one I'm going to set to on. It's now an incommensurate feed-forward loop, but it's actually in space. So this one should initially turn on, but then as this longer path allows it to turn off, it should turn off, and that's in fact what we see. And we can also do, and I think the most interesting thing we've studied, we can make, this is the same network I just showed you, but I reshaped it, right? G1 turns G2 off, but now I've made it, and this is an oscillator. If I make it this way, if I place these nodes this way and I don't freeze G1 to be on, now I just let them all go from some initial state, I'm going to get an oscillation, and that's actually what I get. So this is, again, COMSOL simulation. And you get some period. This makes an assumption that everything has the same rate. There's a couple of interesting comments I wanted to share. So one is that if you tune the spacing between these, you change the period, which is interesting. But also if you put these all, if you make the spacing 0, they do not oscillate. And that has to do with the fact that you need the sort of separation of rates and sort of times, right? You need a delay to get these oscillations. And in this case, this is really hard to do biochemically in general. There are very few true biochemical or even chemical oscillators. But by incorporating the sort of transport, right, you tune the delay with your geometry now. And I guess the other comment that we haven't looked into is that we could obviously start making coupled oscillators. I think there's lots of interesting problems in this space that we have not played with, and we're working on building some of these. All right. So back to, I showed you some toys, but I really didn't tell you very much about this, although I hopefully told you that we can take molecules, and we can do this with multiple, we can have multiple of these elements that each produce the same message, in which case their outputs are added. So I'm going to go back to this notion that in some sense, this is a type of each of these elements. I can put even a network in a single post as a feed-forward gene is sort of some artificial neural network, and actually doesn't need to be feed-forward. I should take that off. And this is not my work, but this is a really beautiful set of work. This is a cellular automaton. And the goal, this cellular automaton on a grid, is that depending on its initial state, it should reach a final color that's dependent on the shape of the nodes that were turned on. And here it's whether the digits, that you form a digit of this shape. And these are about a few, like maybe 50 or 60 nodes, right, that are on in a larger grid. And this is a video of how it works. You can see that as you're drawing, right, the nodes are interacting and they're making a decision about what they most look like, and it can change, right, because it changes depending on what the signal looks like. And this is really fun. You should definitely, as I said, now I work. But But it can also do this in tandem, right? It's sort of local, that it's talking to its nearby neighbors. So you're like, okay, so what's the CA? Well, these are learned, right? So you just give it what you want as a condition, and the idea is that this cell has 16 channels. It does some simple linear transformation on those channels to get some outputs that tell it what to send next, right? And so the idea, so you could actually learn this by training a neural network, I think in this case a convolutional neural network, which you would guess by this, to do this. But what I want to say is, this is what we were doing, right? I have 16 channels, right? I told you I have 20. I haven't built them all at once, and I can make a neural network. So anyway, so that's where we are. I want to add one other point, which is that's a sort of discrete version. I guess we could think about this in some continuous version, right? And I haven't thought about this yet. So that's what I have to say, although I think this lends more questions than answers. First of all, I don't know how close we are to actually building this type of object. I'd be happy to talk about it. I feel like one comment is that our cycle time is really slow. This one's been slowed down, but it is on the scale, so I promise you hours. I don't know if we can do this. And I feel like our question is, can we do this faster? And one comment is the analog state of biomolecular networks is really interesting. So I don't know if this is worth thinking about. And then the other thing is, in all my simulations, every cell worked perfectly. It was all the same. If they're not really the same, how well is this kind of strategy going to work, and how do we fuss with it? So those are my open questions. But what I told you today is that, how do we think about sort of synthetic cell communication? We can think about this notion of localized biomolecular reactions and simple computations as a way to make distributed decisions. And if we can do this, maybe we can build some useful robots. So that's what I have to say. Thank you very much for your attention.